Teams meetings because of the earlier sunset, but we thought that we'd try something different and have sort of a smaller, more intimate gathering with the presentation also available for people to join virtually. So thanks for joining us to test out this little change in format, and we really appreciate your attendance. I was recently very fortunate and invited to assist now Professor Tyler Faith from the University of Utah in some fieldwork he was doing in the Kango Valley. And that's where I met Alex, who was also doing some fieldwork in the Kango Valley. And I asked her if she would very graciously come and talk to us about her work, and she agreed. And so with that, I would like to introduce Alex, and please let's hear what you've been doing.
Awesome. Thanks so much, Jenna. Thank you all for coming. So to sort of explain a little bit of
Someone has said, here we go, done.
I don't know how to make that go away.
You go on the straight line, you click on the straight line.
But my cursor is behind it. Do you see? Like I can't, if I click it, do you see?
Yeah.
Switch out of that back into the next one.
The other one, still.
Admit.
Got it.
There you go. I'm just gonna have to hide it over there.
Okay.
Fine, I'm 10 years old.
What am I doing now? Okay. So I'm a postdoc at the University of Utah. And the work that I'll be talking to you about today is some of my PhD work and now some of the stuff that I've moved into, having finished the PhD. And everything is broadly framed around questions about extinction in South Africa that are, sorry guys, that are related to climate change in the last 40,000 years. And what I specifically do is look at the isotopic composition of herbivore teeth, which might sound a little intimidating, but I'm going to break everything down to explain why this is a really good record that tells us a lot about environmental change in the past. So, over the last 100,000 years, there's been a huge number of extinctions of megafauna all over the earth. And some of the ones that you might be most familiar with that are sort of very famous are mammoths in North America that go extinct right around the time that humans get there. And this has been the case in a lot of places that we have a lot of big animals going extinct right as humans arrive on the scene. So a lot of people have hypothesized that these extinctions were caused by humans, or perhaps that climate change and human activity worked together to cause these extinctions that have impacted the entire globe over the last 100,000 years. But for a long time, people have thought that Africa was not affected by extinctions in the same way that all of the other continents were. And that's because there's still a huge diversity of mammals here, many of which you are familiar with, of course. And so this large diversity of mammals today has led people to say that maybe extinction wasn't a problem here in the way that it was elsewhere. So you can see here from this map, this is a heat map with species richness of large herbivores. So we're saying large is over 44 kilos. And these brighter colors are where we have more diversity of those mammals. So you can see compared to everywhere else, Africa has a really, really high amount of diversity. But that present day diversity doesn't mean that extinctions didn't happen. Diversity in the past was much greater than today. So these are some images of some now extinct African mammals, but we can think about what landscapes might have looked like with more of these mammals on them. At some sites 4 million years ago, we have up to 5 different types of elephant species living in the same place. And in other sites, we have up to 12 species that are over 1,000 kilos. So we used to have a really, really large number of really large animals on the landscape. And
Can you adjust your microphone just slightly to be a little bit more sensitive? Some of the guys are, their audio is just a little bit, sorry to interrupt. There's a little setting on your mic that will.
On your computer laptop mic?
sorry, guys.
Some are hearing you perfectly and some struggling.
Someone also told me a few days ago that my accent is unintelligible. I don't know how much that has to do with it.
Would it be?
We can hear you fine.
Yeah. There we go. That
Is
should,
it this one?
yeah, I think that should be it.
Yeah.
Okay, great.
Okay.
Now, let's see.
Yeah.
Okay,
We're back.
so we do know that we had all of these African extinctions because the landscape today looks really different than it did in the past. But these extinctions are really understudied. So part of the goal of my broader research agenda is learning more about these African extinctions. The specific time period that I work in is called the Pleistocene-Holocene transition. That happened about 10,000 years ago. And during that time, we see the extinction of 7 grassland herbivores, which were, you're probably familiar with some of these, the longhorn buffalo, the Cape zebra, the Bond springbok, and the giant wildebeest. These are some of the species that went extinct about 10,000 years ago. And these extinctions are associated with this period of climate change where we have, perhaps you've heard of the last glacial maximum about 20,000 years ago, which was this very cold period, into the start of the Holocene about 10,000 years ago. We have continuous warming. So people have hypothesized that that warming is causing environmental change, and that's causing the extinction of these grazers. So the question that this raises is, are the changes that we're seeing in the herbivore communities, who's there, which animals are dying out or leaving the area, are those changes in community caused by changes in environment that are really related to the amount of grass that's available to these animals that prefer to eat grass? So the way that we can try to figure out what's happening with this is by looking at who's present at different sites across the country during this time, how those presences or absences are changing through time, and how records of the plants that are available are changing either in sync or out of sync with those faunal community compositions. So, to introduce you to the study area, this is a map that you'll see often when people like me talk about climate in Southern Africa. This map is showing you the proportion of winter rain across this part of the continent. So winter rain, June, July, August. The darker colors, your blues and purples, are higher proportions of winter rain. Your yellows and greens are your higher proportions of summer rain. So as we all know, just from this weekend, we are in this area that's dominated by winter rain. The region out to the east and to the north is dominated by summer rain. And then we have this sort of teal in the middle where we get 2 rainy seasons. So when we think about climate change in Southern Africa, it's complicated because we're not talking about one climatic regime that's changing. We're talking about multiple different systems that exist, all being affected by something like global temperatures rising, and potentially all being affected in different ways. So this is the broader framework for considering what's going on. We need to understand all of these different regions and how they're changing. So these points on the map are colored by the region that they're in. The red points are our winter rain. The blue points are our summer rain. The orange points are our year-round winter and summer rain sites. The shape of these points is telling you what type of data it's giving us. So the circles are archaeological sites where we have mammalian communities that we can look at the composition of. The triangles are sites where we have pollen data. So pollen records, I think that someone just spoke about pollen records to you guys recently, right? So not to talk too much on that subject. We have sediment cores from these locations that have pollen recorded in them. And that pollen can tell us what type of vegetation was growing in that region at that time. So what we want to do is compare these community records of animals with these pollen records to see if changes are happening at the same time. When we think about communities, we're thinking about these categories of animal that are defined by what type of plants that animal likes to eat and how much water it drinks mostly, because some animals need to drink a lot of water, some don't. Some animals prefer to eat shrubs or leaves. And so this is the sort of like sciencey complicated version defining these categories, but really we just have browsers, which are our woody plant leaf shrub eaters, grazers, which are grass eaters, and mixed feeders, which eat both. And then we have omnivores, which eat everything. I try not to talk about them too much because they eat, like they'll eat trash. They're not a helpful indicator to us in this instance. So we're thinking about these different categories of animals and who's present at these sites through time. And then to make our time intervals more discrete for comparisons, we generated these bins. So we have before the LGM, the last glacial maximum. Before the last glacial maximum is our first bin, which is this period of cooling going into the last glacial maximum that I mentioned earlier, which is this very cold period. And then after the last glacial maximum, we have what we call the LGIT, which is just this transitional period of warming from the last glacial maximum into the Holocene. And then the Holocene is much warmer in general than the preceding periods. So we're going to use these bins to compare our fauna and our pollen. What I'm showing you here is a detrended correspondence analysis that I don't need to get into the math of at all. What I want you to take away from this picture is that this graph is separating out our points, which are our faunal communities through time, based on if there are more browsers, more grazers. These points are colored by the time period that they're in, and the text represents the names of the different sites that you saw on that map. The names are not so important. What's really striking about this is that you can see the distribution of colors immediately on this graph, right? So we have our blues and purples over here, which are which are our older units, and then our oranges and reds on that side, which are our younger units. And this side is where our really grazer-dominated communities are. That side is where our browser-dominated communities are. So older sites have way more grazers, younger sites have way more browsers. Here's another way to visualize that. This curve through time, yeah, a little bit easier to look at is this curve through time is showing us our proportion of grazers. We see it go up and then plummet down in the Holocene as it's getting warm, right? This huge drop in grazers makes us think that there's probably a huge drop in grass, right? That these grazers are disappearing because they don't have grass to eat. But when we look at the pollen records at the amount of grass pollen that's in those records. In the same way, we see something really different. The amount of grass at individual sites can be pretty variable, but through time, we're not seeing a decline in the amount of grass on the landscape, or at least the amount of grass pollen that is recorded at any given time, which is really interesting. And not what people have thought is going on for a long time. I have one more in case there are any stats nerds here. I'm okay if I'm the only one, I'm used to it, but just in case, we do have significant differences in the amount of grazers between our time intervals and not in the amount of pollen. So this is again telling us something really different is going on with the grazers than with the amount of grass, which is really contrary to what we would just logically expect. So this raises this really big question of what is happening to the grazers? What is happening on the landscape that the grazers can't live with if it's not a change in just the amount of grass? And this is where seasonality becomes really important, because the time scales that are recorded in our pollen cores are averaging out all the variation that happens within a year and within series of years. So we're not picking up on the changes on the landscape within a year that do matter for animals, right? The difference between when it's rainy and there's a lot of fresh vegetation, when it's dry and there's none, that's really dramatic for an animal on the landscape. And we don't know from these pollen records if the change in grass is seasonal and not being recorded in our pollen cores, or if there's something else going on. So this is why I'm really interested in seasonality, because it's this archive that's really hard to access, but is really important for what animals actually have to do to survive on a landscape at any given time. To think about how animals respond to seasonality, to seasonal changes in the amount or quality of plants that are available to them to eat. They either have to move, this is a picture of the wildebeest migration. They either have to move, go somewhere else to find food, or they have to change what they eat. So some animals, like the springbok, will seasonally go back and forth between eating more grass or eating more leaves and shrubs to adapt to what's happening. So animals kind of have these two options, or to just die, which is the worst option, obviously. So we want to understand what types of decisions animals are being forced to make during these periods of climate change, because that will tell us if we're seeing seasonal change in grass, what's causing extinction potentially. The problem accessing seasonal cycles is sort of, as I was mentioning with pollen cores, a lot of the proxies that are available to us in the fossil record are averaging huge amounts of time. So pollen records are averaging across a large amount of time, other sedimentary core records as well, which is, can you see the cursor? Which is kind of what you're seeing here, that most of our scales of evidence are capturing really large amounts of time from 1,000 to 10 million years is what is available to us in most records. When we're talking about seasonality, we're talking about within a year. So this is a really challenging problem to try to address in the archaeological record of how do we get at that really, really fine scale when this is generally what's available to us. And now we get into why I'm pretty obsessed with teeth. And that's because herbivore teeth provide a really unique time scale that is not available to us in virtually any other archaeological record or paleontological record. These are zebra. These are their teeth. And what happens when an animal eats is that the chemical composition of all of its food is recorded in its teeth, in all of its tissue, and your bones. This is true for you as well. Your bones, your hair, your skin, all of it is recording the chemical composition of all the food you eat and all the water you drink. So teeth are really exceptional for us as archaeologists because they are extremely hard. Your teeth are way harder than your bones. So they preserve in the archaeological and fossil record pretty well. They can sort of take a lot of damage and not fall apart in the way that a lot of other materials don't preserve archaeologically. Teeth are also really cool because they're sort of one and done as far as growth goes, which your dentist has probably told you at some point that your bones can repair themselves if something happens and your teeth cannot. Your teeth are growing and capturing a really specific moment in your life, and then they're never remodeling or overprinting on that signal. So a tooth captures the chemical composition of what you're consuming at a very exact moment. The way I always phrase this is, you are literally what you eat. It's not just an expression. Everything that you eat is recorded in your tissues. This is where I turn into a chemist for a minute and talk about isotopes, so bear with me. Our question is, what does your tooth enamel, that part of your tooth that's growing and is really hard and is never regrowing, what is it actually recording? This is the periodic table, which may ring a very distant bell from when you last took a chemistry class. Don't freak out, we're going to just zoom in on carbon for now. So carbon is the element that everything is made of. You know, we often hear Earth referred to as the carbon planet. Carbon is what makes up everything. And there are three carbon isotopes, but there are two of them that we want to talk about right now, because these are the two that appear in different proportions in different plants that animals eat. So an isotope is functionally a version of one of the elements that we see on this table that is heavier or lighter, but the same size. The way that I like to visualize this is to say we can have a bowling ball and a beach ball that are the same size, but very different weights. And that's a good analogy for isotopes, that we have these two elements that are the same size, which is how we define an element like carbon, but are different weights. And in plant foods, there are different proportions of these different weights of carbon. So we can look at that proportion and say this was a grass or this was a shrub, and figure out what an animal was eating in that way. The way that we connect this to seasonality is that mammal teeth, including yours, this is supposed to be a schematic of a human tooth, grow in layers. The simplest way to think about this is that they grow like tree rings, that your teeth are laying down layers over time as you age, as you grow them when you're a child. And so in these layers, we're capturing different moments in the lifetime and in the diet of the animal that's growing that tooth. So we're changing the ratio of those isotopes through time, and we're recording them in the layers. Now the question is, how do we get that data back out of the tooth? How do we get that information back out? What we can do, this is a zebra tooth from that extinct Cape zebra that I showed you a picture of before. I always think that I was born in the right generation, because imagine having lived at the same time as this enormous zebra. This tooth is like 8.5 centimeters long. These things were enormous. Like, if I saw one of those, I would be out of here. But we can look at a tooth like this. This is from the archaeological site Boomplaats, if people have heard of that site. And we can look at the enamel's isotopic composition at various points down the length of that tooth, sampling different layers to reconstruct through time fluctuations in diet, and then extrapolate that into changes in environmental condition. So this is in a perfect world, we sampled this tooth, we get our isotope curve, and then it perfectly matches what's going on in the environment. Unfortunately, everything is more complicated than that. And so we have to do a fair amount of work to get from that isotope curve to our environmental curve. I'm going to talk about oxygen isotopes now, but we're sort of tracking the same idea as carbon, that your oxygen isotopes will change as your environment changes because they're related to the water you consume. So oxygen isotopes change if you're in a rainy season versus a dry season. So we can theoretically track those rainy versus dry seasons with oxygen isotopes. So this zebra is drinking water in the place that it lives, and that is creating this oxygen isotope curve that is kind of wiggly because nothing's perfect, and it's drinking from different water sources, and sometimes it rains more intensely and other times not so much. So we get this wiggly curve that represents the water that that zebra is consuming. When it gets incorporated into the tooth, you can see that the shape of that curve changes. And that's because that beautiful tree ring schematic that I showed you before is kind of a lie. Teeth don't actually grow in that exact way. They grow similarly to that, but the way that enamel forms is more complicated. So we're changing the shape of that wave and smoothing out some of the variation when we record it in the enamel. And so I want to walk you through some of why this is so complicated. That might be a little small to see in the back, and I'm sorry about that, but I'll try to explain so you don't need to see so much. This first, or I should say, your enamel grows at different rates and different angles through time. So instead of just having these layers that are laid down, the rate of those layers growing and the angle that they're growing at is changing. So you can see sort of immediately how that schematic breaks down, that we're not just capturing like same increment of time, same increment of time, same increment of time in the layers. So these three colors represent the first, second, and third molar of a zebra. So first is teal, second is yellow, third is purple. And the y-axis on your left, just checking, on your left is showing you the decreasing growth rate through time. So your enamel is slowing down as it grows. And that means that you're averaging more time and more change into the same amount of enamel later in the tooth growth than you are earlier in the tooth growth. Earlier in the tooth growth, you're growing fast, you're recording just a moment, and then you're on to the next segment of enamel. Later in the tooth, it's growing slower, you're capturing way more time in any specific piece. The other y-axis here is the angle that the enamel is growing at. So from that sort of center of your tooth to the surface, there are multiple layers of enamel that we can imagine. And they're growing at an angle so that the speed is different between the layers, And so you're capturing different moments of time between the inside of your tooth and the outside, and between the top of your tooth and the bottom. This is just a sort of graph to show you the consequence of that changing growth rate through time. This is tooth length. So you can see that it starts off increasing quite rapidly, then kind of tapers off until it reaches the maximum length. This figure is showing you the amount of enamel that has grown for any specific part of the tooth. And enamel grows in sort of two phases. So first, you get the skeleton of your tooth. That's what's growing in these first two figures, is just the skeleton of your tooth. And that's about 25% of the actual mineral content. Then this second wave comes through and deposits the rest of it. And so now you can see this is getting even more complicated, because we're getting some of the isotope value from that first phase where the skeleton gets laid down, and then more isotope value from a later time where we're getting more of our mineral content. And then to just make it a little bit more complicated, that secondary wave that comes through is at a different angle than the first wave of growth. So we're getting a lot of different moments of time averaged in different pieces of enamel. So this ends up being very complicated. And I want to show you this video that's of the enamel mineralizing in a zebra tooth as a model, that gives you a sense of how this pattern looks in real life. So you can see it slowing down as you get towards the end of the tooth, that angle changing, and that the first angle of the edge coming out is different from that second angle coming through. So we're getting a lot going on in 102 weeks here. And this is what's causing this problem. We're averaging through time in all of our little samples of enamel, and that's causing us to lose variation in the original input data. But we can build models that allow us to recover the signal that was input. So what you're seeing here is that light blue line, that's the original input, behind a dark blue line that is the model we can use to take those red points and turn them back into the pattern that we had as input. Now I'm going to show you a little bit of math, because I had to learn matrix algebra to figure this out, and I'll never stop talking about it now. So do not be intimidated by this math. What this math is telling you is that we've built an equation to say, what is the isotope value at any given moment in time, and what percentage of that tiny piece of enamel is growing at that time. So it's really not that. It's sort of a simple idea underneath all this math, right? That if we know what amount of enamel is growing at each moment in time, we can say this amount of enamel had this isotope value, and this amount of, so 0.25% of this piece of enamel had this value, and then another point however much percent had this, and another point however much had this, and all together that gives us this value. So that's what we're doing here. We know how much mineral content has what isotopic signature at each time increment. And if you're sweating already, the next math is much worse. I'm not gonna make you try to understand this math, because here we're using some some matrix algebra that took me many months to learn, and we don't have time for today. But what I am showing you this for is to say, once we have this model and this equation to figure out mineral content and isotope value being added to any given cell, we can use a baseline value for the tooth, say, okay, across all the values in this tooth, the average was this. Let's use that mineralization model to backtrack and say, what's the most likely input curve that created this isotopic signature in the tooth? And this is called inverse modeling. And the inverse model can help us recover that input signal much more faithfully than just looking at those points of data that we get when we drill the tooth. So what you're seeing here is what you're seeing here is the way that the inverse model works on different inputs of original signal. So underneath each of these, you see our light blue line, which is our original input, our black points, which are what we would measure if we just ding, 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 drilled down the length of the tooth and looked at the isotopic composition of each of those pieces of enamel that we drilled. And that's scaled to the top x-axis, which is tooth position, which is just where on the tooth you took the sample. So we're going from the cusp, the top of the tooth, to the bottom as we move from left to right. And then our dark blue is our recovered signal. And that's time in weeks on this bottom x-axis here. And if I do say so myself, this is a pretty good model. We can recover a lot of that original input signal through these equations by going through these steps of figuring out how the tooth grows and then backtracking. We can get, especially in the first 50 weeks of tooth growth, a really good estimate of what the input signal was. And it's sort of interesting to look here at the differences in the length of the seasons that we're recording. So this one is a 6-month periodicity. The second one is a year. The third is 13 weeks. We can see that we're losing the most amount of variation in 13 weeks. And that's because those seasons are happening so quick that the averaging that's in the tooth is muting them still. The longer our seasons are, the less of that muting we get. That's caused by how much time is averaged in a single piece of enamel. The reason that things get iffier after 50 weeks is because, like I mentioned before, that's when tooth growth starts slowing down significantly. So we're averaging a lot more time in each piece of enamel. We're losing the fidelity of that signal. But it's really exciting that we're able to do this because I want to show you some real world tooth data now. These are a couple graphs of a zebra from Kenya that I sampled, and you can see those black points are the original values that I measured, just plotted on that, where on the tooth we got them from, top x-axis. And you can see that it's hard to pick up a fluctuation there that actually means something in that original data. But then when we run it through our model, we start to see these actual curves, right? In weeks that tell us when it was raining, when grass was changing, that this zebra was affected by it isotopically. So what does that mean for the fossil record? Now that we have this model, we can go back and we can sample fossil animals and we can access this scale of weeks in a year that things are changing that has previously been really, really hard in the fossil record. So this is really exciting in terms of our ability to see weather functionally in the past in a way that we haven't been able to before. And so the thrust of this bigger project of thinking about how grass availability within the year rather than grass amount total might be driving this extinction in the Pleistocene-Holocene transition about 10,000 years ago. We can come back to those same sites where we looked at the changing composition of animal communities through time and look at the isotopic values changing within teeth through time. So what does the seasonality pattern in a zebra from 20,000 years ago look like compared to one from 9,000 years ago? And actually pick up changes in seasonality. So we're looking at these three species now, zebra, eland, and springbok. And this is because they all kind of do different things. Zebra are grass eaters, and eland and springbok both sort of change their diet according to what's available. But they do that in very different ways. Eland are more likely to migrate. Springbok are more likely to just change between eating shrubs and eating grass. So we're trying to cover all of our bases to look at a community as a whole and see how the changing seasonality in these different animals lines up and what that means for the landscape. And so theoretically, we will shortly have a picture, and this is where I have to confess that this is a trap to get you to invite me back because I'm still processing this data, so I don't have the results on the seasonality patterns that we're measuring in these archaeological teeth yet. But what will be really interesting about this is that we'll be able to look at these sites in these different regions. And this is the same rainfall map I showed you before, just with a different color scheme. We'll be able to look at these sites in these different regions and identify changing seasonality through time, and also identify differences in changing seasonality between these areas that we already know have dramatically different seasonality patterns today. And this has big ramifications for what climate change means in South Africa, because sort of going back to that initial introduction to rainfall patterns in South Africa, we're not dealing with sort of one type of climate. We're dealing with these very different climate patterns. So understanding how climate change impacts these different areas differently helps us understand much more of the fossil record than just the Pleistocene-Holocene transition, and also has some ramifications for how we think about climate change affecting this region in the future. So this is all of these components coming together to think about extinction and what that meant in the past and what climate change means for Southern Africa broadly. So I think I'm at about the point I wanted to be to switch to questions. I particularly like questions about math. I'm open to all sorts of questions. So thank you all very much.
Cool. Thank you so much.
Thank you.
Firstly, we'll give the room a chance, and then we'll see if there's anyone online that has a question.
Yeah.
I've got a question. So it doesn't matter between to male and female animals of the types that you're sampling, you know, the springbok, for instance, you know, their behavior within their own life way. So, you know, male springbok behaves quite differently to females that are young. So
Yeah.
in your, how do you differentiate between them, and then what do you expect in your graph, in your model?
Yeah, that's a really good question. I'm particularly interested in this with eland because springbok mostly stay relatively local, but eland can migrate so far, and the females and the juveniles have such different patterns than the males. But what's important to remember is that your teeth grow when you're young. So we know we're mostly capturing that juvenile pattern, which is helpful since in the archaeological record, we're mostly dealing with isolated teeth. We don't get a lot of the time enough of the animal preserved to say this was a male or a female. We'll be like, hey, great, I found one springbok tooth. So we don't have that level of resolution on the fossil record, unfortunately, a lot of the time to make that distinction. But it is the juvenile record that we're capturing. So for example, in zebra, the first molar grows from when the animal is in utero to when it's a year and a half. And then the second molar grows from about a year to about 2.5. And then the third molar grows from 2 to 4.5 or something like that, approximately. So we do know what section of the life of the individual we're capturing, but I am really interested in this, particularly with eland, and someday as a side project, I'll do more with that. Yeah.
The difference between eland and the springbok. Springbok are historically to migrate.
Yeah.
Numbers, which of course we start out and do. But
Yeah.
my question is, have you considered, and most people working in herbivores don't consider the need for salt, and where does the salt patterns influence migration, and would you be able to use the sources of salt? Of course, the course you have plenty of salt, but once you're moving inland,
Yeah.
there are certain areas, some rivers are saltier than others. Would that have an effect upon the migration and therefore your model?
I don't think that changes in migration patterns would change the way to design the model, but I do think one of the things I've just started looking at is gradients of plant palatability and nutrition across things like altitudinal differences, going up and down in elevation, thinking about how this changes, and also looking at the soil and the components of the soil that I think feed into this idea of what amount of salt and cations and nutrients are animals getting in different locations. I think will contribute to a better understanding of that. But I haven't looked at that specifically yet.
Any other questions in the room? No? Nick, any online?
I've asked them to post their questions to the chat or to put their hand up. So if someone puts their hand drum roll. So we have 11 people on, well, we had 11 people, now 9. So we've got Jeanette's still on. She managed to sort out her audio. She had the microphone, something about microphone.
Okay.
Okay, good.
Let's see.
Okay.
Should I give them last chance?
Last chance for questions.
Yeah.
I just love it. So are you also like looking at the contemporary tooth records at the moment, and are they matching your model that you like very closely, or like you have to make some adjustments?
Yeah, so the model is all informed based on modern
Okay.
tooth data. So we're looking at, I should have included a picture. What we look at is a thin section of the tooth, and we can see sort of those layers. And a specific detail about the way that tooth growth happens is that there's a tiny amount of enamel that's laid down every day. And so you can count those daily lines and the way that they're spaced between layers to understand how fast and at what angle growth is happening. So we're getting that data from modern animals, and we're collecting modern data from across Southern Africa, from Western, Eastern, Northern Cape Freestone, trying to cover all of our bases to characterize what's happening in these different modern ecosystems really well, and to test the model really vigorously against what we know exists. And I didn't include it because I didn't think I'd have time, but this Kenyan data is from a place called Olpejida Conservancy in central Kenya, which we chose because a colleague of mine had a bunch of teeth, first of all. But, well, she had the whole zebra, and I had to pull the teeth, which was astonishingly difficult. But the reason that we wanted to look at those zebra is because Olpejida is a conservancy, so they have measured rainfall amounts for the last 15 years. So we could look at the seasonality patterns of the rainfall really specifically, and then look at the seasonality patterns of the teeth and correspond them. And so, yeah, so we're basing this all in the modern and making sure that it actually works. And it does work. I should have included, we looked at sampling multiple teeth from the same individual, and when we create a sequence of multiple teeth sort of stacked together, we pick up this really, really nice bimodal precipitation, which is what we see in central Kenya. So it was really exciting to see that the model is working so well on actual data that fits with what we're measuring in rainfall. So we're working on that in modern South Africa now, where I'm needing more time because I did not conveniently have a friend who already had a bunch of dead animals. But we're working on that sort of in tandem with the archaeological, so that we can compare to modern and also make sure the models are working the way that they should.
You can tell Tim to go ahead.
Yeah, Tim, go ahead.
Thank you very much. Very interesting. So I'm not, I don't know much about the archaeological record, but I work mostly with contemporary ecology and maybe historical ecology. And you've partly answered my question already in the previous, your response to the previous question about contemporary data. But my area of interest is actually in the Karoo, where I know that over the last 100 years or so, there's been a shift to more grassy conditions, coincident with a reduction in livestock numbers, but also with an increase in more summer rain. And some of the early literature about the Karoo would argue for distinct summer rain would drive grass cover, and winter rain would drive shrub cover in the Karoo. So it wasn't just about rainfall seasonality, but also about the response of the vegetation to that seasonality. And so the C3 shrubs would respond to cooler winter rains, and they sometimes occurred in several years in succession. And then the warmer summer rains, which has been the predominant pattern over the last 30, 40 years, would result in an increase in grassiness, which is what's been happening in the eastern part, that ecotone between the grasslands and the shrublands. So it's a very specific area. I don't know about Boomplaas and what would be going on there, but just I wanted, maybe you could comment on dealing with this kind of contemporary plant ecology or rangeland ecology literature as well.
Yeah, delighted to. So what Tim was just introducing is that C3 and C4 plants, our C3 plants are generally woody plants, and our C4 plants are generally grassy plants. They favor different growing conditions. So C3 plants do a lot better in cooler growing conditions, and C4 plants do a lot better in warmer growing conditions. So in places that we have winter rain, we see a lot more C3 plants, and in places we have summer rain, we see a lot more C4 plants. And that generally means that places with summer rain have a lot more grass, places with C3, or with winter rain, have a lot less grass because it's mostly C3 plants. There are C3 grasses, but there are comparatively very few C3 grasses compared to the number of C4 grasses that we have. This gets really interesting when we get into this zone that gets winter and summer rain, because when we're talking about amount of grass, we don't necessarily know from a pollen record, for example, if it's more C3 or more C4 grass. We can't determine that. And so we see, for example, now an increase in summer rain leading to an increase in C4 grass. But when we look back to, for example, the last glacial maximum records at Boomplass, we see an increase in grass that looks like it might be an increase in C3 grass. And so this is why this sort of intersection zone is so interesting, that we really struggle to attribute what type of grass to what season in many ways. And partly why zebra are interesting is because they only eat grass, and C3 plants have a very negative carbon isotope value compared to C4 plants. So if we look at a zebra and it has a very negative isotope value or isotope profile in its tooth, that's telling us about the presence of C3 grass. And in certain time periods, we might expect C3 and C4 grass to be coexisting, and their relative proportions increasing and decreasing seasonally. So that's something that we're trying to understand and characterize, that that is a function of sort of changing incursion of summer versus winter rain, like what we're seeing now in the Karoo. Does that get at what you were mentioning, Tim?
Yeah, very much so, especially because like Boomplass is at this, as you say, this transition between winter and summer. And when it's lots of winter rain, you get more shrubs. But generally, over the last 100 years, there's been an increase in shrubbiness across much of southern Africa, bush encroachment and all that, which of course would drive an increase in browsers. So that would be consistent with your pattern of an increase in browsers over time as well.
Any more questions?
Can't see any.
Okay. Well, then with that, thank you so much for coming, Alex. It's been lovely having you.
Me.
I think I learned a huge amount. You managed to simplify what could have been incredibly complicated. So
Okay, I'm glad no one got up and left when I got the periodic table or any math.
You guided us through it like a superstar. Yeah, thank you so much.
Yeah,
And, we will definitely have you back. Yeah, you will. Thanks, everyone.
Thank you.
Thank you.
I don't think any zebra ever thought his tooth was.
So valuable.
No, I don't think so either.
Tooth double that.
That and more.
So the